Hello, hello,
Can I say to check the voice from the
hello,
students?
hello, hello, hello,
It's on now.
Good evening, my dear students. Due to some issues of net, there is a delay. So, hope so. Now, we are going to start with a good beginning. And last lecture, we discussed regarding morphology of flowering plants. I hope so you understood that topic and you have revised that as well. Now, today lecture, we are going to start with the anatomy of the flowering plants, right? Earlier even I said you in my class that morphology are of two types, internal as well as external, right? In last lecture we discussed about external morphology. Today we are going to discuss about the internal morphology, where we are going to take first under the same chapter histology, then we will discuss about anatomy. Actually, to reach up to anatomy part, you should have good knowledge of histology. I hope so you know now the histology. Let's recapitulate. Please give me the answer. Earlier I said you the, what do you mean by the term histo? Please give me the response. Okay. Volume is already increased. Please do check the volume again. Is that audible now? Audible now? Okay, answer is correct. You have given the answer as a tissue, right? So here histology means tissue, right? Very nice. Means that is audible. That's why students are giving the response, right? Now please put the thumbs up if everything is going fine now. Give some response. Okay, fine. So we are getting all the clarity in the voice as well now. Answer even you are giving me. Very nice. Last time we discussed the word histo stands for what thing? Tissue. Ology stands for to study. Clear? So first of all we will discuss about the tissue. Then we will enter into the anatomy part. Right? Now to learn a little bit about what is anatomy. So for this thing we are having this part. Can you see the cells which are arranged into a tissues, and tissues are constituting some organ. Means if we are taking some organ of a plant and learning the arrangement of the cells there, that you call it as what thing? Anatomy. Are you getting that? So what is anatomy? Study of internal structure or organization of plants. As we are dealing in which anatomy? Anatomy of flowering plants. Like last time I said you, morphology, physiology, these are even common terms to both plants and animals. Even the histology and anatomy are the common terms to plants and animals. But we have to specify which organism we are discussing. We are discussing here regarding the plants. Fine. So again here some students are putting that some volume. Please do check your any of the device from where you are following this video. Please do check your device as well, because major students are putting thumbs up, so it is going fine. And if somewhere it is missed, you can watch the this video later on the YouTube. Fine. Okay, now see here, this is about the concept of the anatomy, right? So anatomy is of any part of plant. If we are taking the gross internal, overall internal structure, that you call it as anatomy. And if we are learning only about the tissues, the separately, every cell separate, right? What they are doing means the different tissues perform different functions. So that tissues when organized together, they create some organ. Are you getting that? So only talking about the tissues is what thing? Histology. Let me to make it little bit more darker in color. So here it will be what thing? Histology, right? The study of tissues, what we call it as? The study of tissues, what we call it as? Histology. Now that different tissues cluster to create some of the organ, and you are learning the gross internal thing, how such tissues have been arranged, that you call it as anatomy part, right? So I think so it is clear to everyone. Now let's see for the next one. Here you are having like this, the tissues how they are arranged. These are arranged like this is a cluster of tissues performing some specific function, and here they are internal performing some specific function. But what is that function? We will discuss in a respective transfer section, right? Transfer section means when we are cutting an organ into the horizontal way, that you call it as transfer section. Clear? So here what is happening? The tissues are clustering and creating one organ, right? Right in front of you is the longitudinal section. Longitudinal means lengthwise section of the root is there in front of you. On that day we have discussed external morphology of the root. Today we are discussing about the internal morphology, means anatomy of the root. Just simply just to show you the organization of tissues, how they are happening inside the root, right? So here just what is the tissue? Can I say it is a group of cells? Just now I have shown you from this slide that what is a tissue? That is a group of the cells, and what they will do? They will perform or having some common origin as well as performing some common function. Two things are important here for a tissue. Number one, it should be having common origin, and next they are performing together common function. Is that clear? Okay. Now after this, what doubt you are asking? Okay. Fine. So please do check it. Please do say that whether it is clear or not just once. Even we are checking this point, just check it. Is that clear, everyone? Okay, it's fine actually. Okay, fine. So we should continue with our topic now. Next, what is there? Types of tissues. Let me to remind you what we are learning. First of all, histology. Then after that, gradually we will go to the anatomy part, right? So now in the histology part, let's see, tissues are of how many types? Basically, two types, meristematic and permanent. Here the term meristo, this meristo means what thing? Divided. Means the cells keep on dividing over here. And where they are present? In a specialized regions. They are present in specialized regions. Region means area of the plant. And that region you call it as what thing? Meristem. That region you call it as meristem. Please listen this point very carefully. What do you mean by meristem and what do you mean by meristematic tissue or meristematic cell? Meristematic cell is what? One part of that tissue, right? And cluster of such tissues, right? Or cluster of that cells, I must say. The cluster of that cell, that you call it as tissue, right? So that region together, that area in a plant where that meristematic cells are present, that you call it as what thing? Meristem. Fine. So what we are discussing? Types of tissue. The first type what we are discussing, meristematic tissue. What is their main thing? That they keep on dividing. They keep on dividing. You know that plant keep on growing, growing and growing. This is all due to which type of tissue they are having? Meristematic tissue. Animals do not have such meristematic tissues. That's why they cannot grow throughout their life. They cannot grow throughout their life. Who? Animals. But plant can grow throughout their life. Just a moment. There is some issues. Okay, sorry for this interruption, it is happening in between. Just please focus back again. Here is what tissues. First, I repeat, meristematic we discuss. Second, what we are going to discuss, permanent. Permanent means the cells of the, or you can say cells of that particular tissue are not dividing. They are not dividing. They become permanent. They are specialized to perform some specific function. Are you getting that? So just recapitulate the main heading over there. What is that? The main heading here about histology, study of the tissue. And what is the tissue? Cluster of the cells. And which type of cells? Whose origin is same and function is same. That you call it as what thing? A tissue. Now tissues we are discussing their types. Number one, meristematic tissue. Second is a permanent tissue. What are meristematic tissue? Which keep on dividing. What are permanent tissue? That will stop dividing and will specialize to perform some specific function, like leaves. They will stop dividing at certain age, right? It will make some shape, structure. After that they will stop dividing, and what function they will perform? They will perform the function of photosynthesis. Is that clear? So these are the two types of the tissues just now we have discussed. Now coming to the next part. Let's take first of all meristematic tissue. Meristematic tissue are of two types. One is based on location, means place where they are present. Second, based on origin, from where they have been originated. Let's see first of all based on location, means the place in a plant where they are present. Like first is what thing? Apical meristem. Let me to show you one slide for this. Okay, can you see here? This is the stem part, and in that stem part, the meristem region is at the upper side. This, which meristem? Apical meristem, right? It is on what side? Upper side. The word apical means on the top, at the tip. If it is present at the tip of the stem, then you call it as a shoot apical meristem, right? Or at the down base, root. If suppose this is the ground and below is the root, on that day we discussed the root cap is covering the region of meristematic activity. So they are having the root apical meristem. What they are having? Root apical meristem. Keep in your mind, the meristem is not throughout the plant, it will be at specific regions. And what are that specific region? Just now I discussed with you. One of the specific region is what? That will be the apex. That's why you call it as apical meristem. And what is intercalary meristem? I hope so you remember on that day we discussed nodes and internodes. See, suppose this is a stem part. Fine. So now see here, what is this part from where the leaf originate? Please give me the response. Last time we have discussed this thing. The part or the point of the stem where the leaf originate. What do you call it as? Please do say that. The point from where the leaf originate. Very nice. That we call it as node. That point we call it as node. And distance between two nodes, that you call it as what thing? Internode. Fine. So intercalary meristem is present on the internode, either below the internode. Can I say this is the internode? So it is base of the internode, or this node is there. Let me to take the color, this one. I repeat, this red one is what thing? Nodes. Clear? Red one is what? Nodes. And blue color, what I have shown you over here, that is the intercalary meristem which is present at the base of the node. Suppose this is an internode, this is an internode. So at the base of it, near to node, what is present over there? Intercalary meristem. It is increasing the length of the internode. Intercalary meristem that increases the length of internode. Are you getting that? An apical meristem overall increases the length of the shoot and even of the root. I repeat, based on location, first we discussed apical meristem, second we discussed intercalary meristem. Intercalary meristem is present at the base of internode. If this is one node, this is second node, the place between is known as a what thing? Internode. And the base of internode, what is present? Intercalary meristem. Is that clear? Now third one, what is that? Lateral meristem. What is lateral meristem? Again to the position. Lateral means they are present on the lateral position. Now where it is present? Let's see through this diagram again. In this diagram, can you see this position? This is on lateral side. Now these lateral meristem, what they will do? These lateral meristem will increase the diameter of a plant. Like have you seen when you are sowing the seed, especially in case of big trees like mango tree is there or any dicot higher plant is there, they keep on growing in a width. Why they have to grow in width? Because they have to support the big length of a stem. So that increase in a width or you can say perimeter or diameter, that will be done by which meristem? Lateral meristem. Clear? Let's recapitulate once again. First, what we have discussed? Apical meristem. Second, what we have discussed? Intercalary meristem. Third, we have discussed the lateral meristem. Fine. So there, when you see a plant, when it is initially, when you sow a seed, how it is growing more? It is growing more in length due to the apical meristem and intercalary meristem. Apical meristem is making the plant to grow in length from the tip, and intercalary helping them to increase in length at the internodes. Are you getting? And lateral meristem helps to increase the width, or you can say diameter, right, or the perimeter of the stem. That you call it as which type? Lateral meristem. So based on the position, I hope so these three types are clear to you. Fine? Okay. Now coming to the part, this back again. So lateral meristem, that increases what thing? This increases diameter of a plant, right? Especially in which case? In case of dicots. It is not in case of monocots. Is that clear? And monocots, usually if I am taking annuals and biennials, their lifespan is very less, because one of the student asked, can we take a sugarcane as an example for the secondary growth? We cannot take it for secondary growth because they are the short-lived plants. The plants which have to survive for longer time, they are keep on growing and growing, so ultimate they need the support through their width. Fine. So that is usually shown, the lateral growth is usually shown in case of the dicots. Fine. Okay. Now after this is what? Based on the origin. Now based on the origin is what? From where they have originate. Like if I say the first is a promeristem. Promeristem, the origin is embryo. You know that after fertilization, from zygote embryo is formed. From that embryo, which already, or you can say that dividing cells which are already there in an embryo, that you call it as promeristem. Pro means from the first, from the beginning. Simply meristo word, what I am telling you, that keep on dividing and dividing. So promeristem means which already is there in the young stage, in the embryo stage, they start dividing from the embryo stage only. That cluster of cell which are making embryo, that you call it as promeristem. Fine. Now coming to the primary meristem. Primary meristem is what? Which is when embryo coming out, it is forming what thing? Shoot system and root system. From the beginning, if meristem still is there, that you call it as primary meristem, right? So this is the first form meristem. And what will be the example of this? Obviously, which type of meristem helps the plant to grow more? Can I say it will be the apical and intercalary meristem in the beginning cases? So can I say here primary meristem example will be what thing? Apical meristem and intercalary meristem, which is from the beginning. That's why your seed can sprout or germinate from the beginning, right? Due to the presence of which primary meristem? Apical meristem and intercalary meristem, right? Then what do you mean by the secondary meristem? Can I say which originate later in the life? Later means what thing? That they are originating later for the increase in the width. So it will be having mostly what of the thing? Mostly lateral meristem. Are you getting that? But here why I have used the word most? This is very important. Please listen this point. Apart from the topic what we are learning, when you are reading NCERT line to line, it's very important to read the words like all, some, most, few, like that. Here I put the word mostly lateral meristem or secondary meristem because they originate later in the life to increase the width of a plant. Right? The width can be increase of stem and even of the root, but in case of dicots, not in case of monocots. Now here which is an exception, due to which I have written the mostly. The example here which is an exception, that is the intrafascicular cambium. What is the example? Intrafascicular cambium. Just learn this word as such, just learn it as such. I will put that word somewhere, then you will come to know. I repeat, promeristem is what? Which is present in the embryo stage where the cells are dividing. Primary meristem is what? Which is first form when your plant just sprouted and keep on growing and growing until and unless the lateral growth is not happening. So that is going up due to what thing? Primary meristem from beginning and throughout the life. What meristems are there? Primary meristem. But later in the life to show expansion, which meristem are there? Secondary meristem. I hope so you get this concept now. Please put the thumbs up for this types of meristem what we have discussed. I recapitulate again primary meristem. Just now I said which is from the beginning. When the embryo start forming what thing? Shoot and the root system. Are you getting that? Then from the beginning shoot is keep on growing upside and root is moving towards the under the ground. Are you getting that? Very nice. Okay. So here secondary meristem for lateral, right? So this is one topic we have covered, and if you are writing, you miss something to write, these videos or these PPT will be saved. You can have a look there. Main focus on the concept what I am speaking. Just understand that first. Okay. Now coming to the next point. Permanent tissue. Permanent tissue, I said you 3 are there, or let me to say from the basic first of all, 2 main categories, simple and the complex one. Now what do you mean by simple? Simple means when all cells are of same type. When all cells are of same type, such tissue you call it as what thing? Simple tissues. And where we are having different types of the cells are there, right? We are talking about the tissue. So obviously from where it have been formed? From the cells. Like we are constructing some building. Think a building is like a organism. So that building, the basic unit is the brick. That brick you are making one by one, making walls. Walls are making rooms. Rooms are making floor. Floors are making the building. In the same way here, cells together form the tissues. Tissues together form the organ. Organs together form the systems. Different systems form an organism, right? So here we are on which level? After the cells, the what is forming? Tissues. Clear? We are on that level. Now I repeat, simple tissues are what? Where all the cells in a tissues are of same type. And what are the types of simple tissue? Parenchyma, Collenchyma and the Sclerenchyma, right? And complex tissue, what are its type? Xylem and the phloem, right? You know that xylem helps in the conducting of what thing? They help in the conduction of water and minerals. Water and minerals. From which part of the plant? From the roots to the upper part of the plant. Whereas phloem helps in conduction of what thing? That helps in conduction of food. Now say me, which part of the plant is forming the food? We discussed morphology last time. Which part of the plant is forming the food? We discussed last time this point. The vegetative part, right? Leaf, very nice. That is the leaf which is forming the food. So can I say the food is transmitting from the leaf to the non-green parts of the plant? Who will be conducting that thing? Phloem. So can I call this xylem and phloem as a conducting tissue? We call them as a conducting tissues, right? Rest of the detail we will discuss later. This is simply an introduction. We have to go with the simple tissues first of all. Parenchyma, Collenchyma, Sclerenchyma, right? So let's see all the types here. Parenchyma, and whatever is written here, don't focus there, simply see this diagram. What is written, I am speaking and explaining you through this diagram, right? Now can you see this diagram is mainly having these cells which are green in color. So parenchyma, its one of the function is what? Photosynthesis. So what cells you will find in a leaf? Majorly it is what thing? The parenchyma cell. So these cells, the function is what? Photosynthesis. In some other places it can store the things as well. Then in some of the cases it can help in the secretion as well. What are that places? That all is a matter of detail of a chapter. Just now a superficial introduction of the topic we are going with. So parenchyma, let's see now, their wall is thin, very thin. You know that plant cell is having cell wall, right? That cell wall here will be thin. And what it is made up of? Cellulose. Fine. Okay, let me to say you one trick to learn the features of this thing, then I will come on this diagram again, right? So let me to introduce that thing. What is that? If I am writing slim cellulosic wall, right? These are indirectly telling you about the features of parenchyma. How the features of parenchyma it is telling you? S stands for its shape, which is usually isodiametric. right? L stands for these cells are living cells, they are not dead cells. We will learn in Sclerenchyma they are dead cells, but these are which cells? Living cells. Next, I stands for they have intercellular space. They have intercellular space. What do you mean by intercellular space? Now see here, can you see two cells in between this white color portion you are looking? This is inter. On that day I said you the word inter stands for what thing? Anyone please give the answer. Inter. I-N-T-E-R. Inter. What do you mean by that? Intercellular, okay. Inter means between two different or within same? In between. Very nice. In between two different. So here two different cells, they have space between that. That's why that space you call it as what? Intercellular space is there over, right? Now see next one what we are discussing here. So here C and M stands for what? This is a major component. This is a major component of plant organs. Means plant organs are majorly form of parenchyma, right? Most of the you can say organs of the plant must have parenchyma there. So it is a major component of the you can say of the plant organs. And which wall it is made up of? Cellulosic cell wall. Are you getting that? So this is a sort of mnemonic. Mnemonic means a shortcut, a trick to learn everything in one trigger. So this is one of the thing that we will teach you how to make the things easy. So slim cellulosic wall. Slim, why we kept the term? Because slim means they have thin cell wall. That's why we use the term slim. Clear? So I hope so. You will go through that thing and must learn the feature of the parenchyma. And what are the functions of parenchyma? Photosynthesis, storage and secretion. Is that clear to everyone? Okay, I hope so it is clear to everyone now. Okay, great. Okay, now coming to the next part. Next we have to discuss regarding Collenchyma. Now Collenchyma, if I show you the picture of this in front of you, I again repeat, don't see what is written, just listen the words what I am speaking through the mnemonics or what, ultimately you will get the meaning of everything here. If some words are still pending, then when you will come, then we will discuss all that points, being it is more complex thing which we should not discuss here. Clear? Okay, so what is there in Collenchyma? Can you see there is no intercellular space? There is no intercellular space, and somewhere there chlorophyll is there. The center part is what? Vacuole. What they have labeled here? Vacuole. What is vacuole? We will discuss later on. Just you should know it is not an empty space. It has some organelle which you call it as vacuole. For that only I speak that word. But what are the main features of that? It is having chloroplast. That's why it is green in color. Fine. And thickenings, can you see it is only at corners? Right? Only at corners, not uniformly on the cell wall. Clear? No intercellular space. They have chloroplast to perform the photosynthesis. And being a thickenings are there at the corners, they will provide mechanical strength. Can you see in the, when the winds are blowing very fast, it bends the stem, tender stems, soft stems, usually bend more. But all are not going to break. They will come back to its original position. This all flexibility is due to this Collenchyma, right? So this is there due to the Collenchyma, and they are having thickenings at the corner. Now how to learn all the characters? I give you one more mnemonic here. What is that? In the name of that, the features are there. Collenchyma, right? C here stands for corner thickenings are there. But that thickenings are made up of what thing? Hemicellulose, cellulose and pectin. It is made up of that thickenings. And O stands for it is observed under, where you observed it, it is observed under epidermis of dicot stem. It is usually observed. When you cut the transverse section of a dicot stem, then you will find this Collenchyma just below its upper layer. What is that layer? We will discuss. So that is on there, and they are living cells, right? They are not dead cells. And obviously corner thickening, even you call it as localized thickenings. You call it as localized thickenings, right? And E here I will take it like it will be either homogeneous or it can be in a form of patches, or patches better I must say. It is going to be in a form of patches, mean discontinuity is there, right? The homogeneous means like complete layer is there. This is usually in the plants, it is continuous layer in dicot. But in some categories like cucurbits, where watermelon is coming, there it is having some ridges. They will go like this, like this, like this. Stem cutting will go like this. Where this depression, this dip will come, at that point this Collenchyma is not present. Are you getting that? Not in all the cases, but in some cases. So in some cases is outside is epidermis. Epidermis means outer layer. We will discuss later, don't take tension of this. But you should know epidermis simply an outer layer. Inside what is there? Immediately which layer is there here? Collenchyma, right? But can you see here it is in a form of patches. So most of the plant it is continuous, but in some cases only it will be in the patches like this. Are you getting that? So here we are having this part. Now M stands for what thing? Here we will take this either like this, and they don't have, just now we say there is no intercellular spaces. There is no intercellular spaces. Now here this C stands for they have chloroplast. Chloroplast are often means usually it is present. And you need not to take for H and Y. I am not taking anything. M and A stands for its function. What M stands for? Here M stands for what thing? Mechanical support. Mechanical support. It won't allow the stem to break when any you can say any bending is there due to any of the reason. So it will not going to break, it will come back to its position. Then A stands for what thing? They help in assimilation. What do you mean by assimilation? Simply formation of food, right? And what is formation of food? Photosynthesis. Why they have to show? Due to the chloroplast. Are you getting that? So you need not to learn the big lines. Just a moment. Okay, so you need not to learn the big lines here, right? So these lines can be put in a shortcut like this. Are you getting that? So please have you understood that part? Okay, A for assimilation, M for the mechanical support, fine, that only. Very nice. So this is about the characters of the Collenchyma. Now coming to the next part, we have to discuss for the Sclerenchyma. Now Sclerenchyma as such you can learn, as the diagram is indicating very nicely this part, but very important is that they are the dead cells. They are having dead cells over there. This is important. First to what we discussed, living. This is dead, and even it is providing mechanical support to the organ. If they ask you the living cell which is giving mechanical support, Collenchyma. If dead cell giving mechanical support, answer will be Sclerenchyma. Now what are its features? Its wall is very thick. Its wall is very thick, and it is made up of which material? Lignin. Let me to write it, lignin. That's why it is written here lignified cell walls. Lignified cell wall means a special structure is there, or you can say a compound is there, lignin. What is that? We will discuss in biomolecules. Just now you should know lignin is present, which is giving thickness to the wall of these sclerenchymatous cells. But in shape they are of two type. What is that? They can form fibers. Can you see it is elongated? Like a boat, it is like a boat, like a ship. It is like this. And some are spherical. But which are spherical, they are much thick. They are having much more thickness as compared to this fibers. Now let me to say one thing. Suppose this is a room, and where you are sitting, that room, if their walls keep on increasing, can I say the space will decrease inside? Here that space you call it as what thing? Lumen. That space you call it as lumen. So can I say due to less thickness, here the thickness is less as compared to this. This is having more thickness. But as compared to other cell, this is having still more thickness as compared to parenchyma, collenchyma. Fine. But compared to sclerate, this is having less thickness. So can I say here lumen is more and here lumen is less. Lumen means space. Are you getting that? So these are the differences between what thing? Fibers and the sclerate. But these two are what thing? These are the types of what? Sclerenchyma. So these are having long, narrow, thick wall. Which material is here? Lignin. Now please answer me this question. Which cell is made up of, or their corner thickenings are there made up of hemicellulose, cellulose and the pectin? Which cells, which tissues we discussed? Please give this answer. Very nice. Collenchyma. One student is asking how dead cell can give the support. Now answer me, how a beam, a pillar of a building giving support to full? It is dead, it is not living. Are you getting that? So it is not a matter whether living or dead, the thickness in that provides the strength, right? So it is not a matter the living thing is there. It is not like us, we are standing with our arms pulled and giving them some of the support like that. It should not be living for a support. The dead things can even support the other things. Like here, the sclerenchyma being dead, still it is supporting the organs of the plant. I hope so it is clear. Who asked this question? Okay. Okay, fine. Now coming to the next part. So what type of cells we discussed? Let's see the previous slide. There we already said the types of tissues, like permanent tissues, three types are there. One is that simple. Under the simple, three types are there: parenchyma, collenchyma, sclerenchyma. I repeat, and the permanent tissue, I repeat, permanent tissue under that simple tissue, parenchyma, collenchyma, sclerenchyma, that we discussed. Now let us look for the xylem and the phloem. Right now, getting for this part, xylem and a phloem, let me to give you some of the things for this. Xylem is further made up of four elements, right? What are that four elements? It is made up of vessels, tracheids, xylem fibers and xylem parenchyma. Fine. Whereas phloem is made up of what thing? Again, four elements. What are that four elements? It is made up of sieve tube cells, companion cells, even the phloem parenchyma I must write first here, and then we discussed phloem fibers, right? Now what is happening in this case? In case of xylem, mostly the elements are dead. In case of xylem, mostly elements are dead. What are that? These three. These are dead elements. Only one is living. What is that living one? Xylem parenchyma. We already discussed parenchyma cannot be dead. It is living. But you remember fibers just now I discussed with you. Fibers come under which type? Please do say that. It will be sclerenchyma, collenchyma or parenchyma. What type it will be? Right. It should be Sclerenchyma. Very nice. That's why they are dead here. So majorly they are dead, basal, tracheid and xylem fibers, but living is what thing? Xylem, parenchyma. Then these sieve tube cell, companion cell, parenchyma, these are living. Mostly members are living of phloem. So only one is dead. What is that thing? Fibers. Keep a formula in your mind to learn which elements are living and dead here. Fibers are always dead, parenchyma is always living. Are you getting that? So if someone asks you which element is dead here, directly say here which dead, vessels, tracheid and fibers. I must say here actually which one is living here. So directly answer will be xylem, parenchyma. And which one is going to be dead in phloem? Phloem fibers, right? So parenchyma, I repeat, will be always living, fibers are always dead, whether it is a phloem or it is of xylem. Fine. Okay, now coming to the next part. Okay, now coming to the next part. These are tracheids and the vessels. The major differences you should know, the tracheids are, these are the tracheids and these are the vessels. And together you call it as trachea elements, right? And these are much broader and there are narrow. They are lumen. Lumen, you remember I said you space in a dead. I said you this tracheid and vessels are what? Dead cells. Clear? Tracheid and vessels are the dead components of xylem. So they have space in between that, which you call it as lumen. That lumen will be narrow. In which case? In case of tracheid. It will be broader lumen in case of the vessels. Are you getting that? So they are like a bundles or pipe, like one pipe linked to the next. Like one member is on the next, then next. They make a complete pipe. So this is a one vessel member, right? Which is compiling upon each other to make a vessel, a pipe. And they are linked to next adjoining pipe with the help of pits. Even for tracheids, they are linked by what thing? Pits. But can you see the difference? It is like a cylindrical in shape and it is going to be tapering in shape. Tapering means becoming narrow at the tips, broader at the center. Are you getting that? Sclerenchyma, beta, it is there, no mnemonic. Simply looking to diagram, 2-3 things you can directly figure out. If you know the feature firmly of first two, then definitely you can search. This odd one is now for the Sclerenchyma. Are you getting that? Okay. Then after this is what? I hope so you get the differences. The lumen will be broader in case of the vessel, but it will be narrow in case of the tracheids. Clear? Now coming to the next part, these are the main conducting. We are not discussing here parenchyma fibers as we have already discussed. We are discussing now the major component of xylem and phloem. What is the major component of xylem? Vessels and the tracheids. Together you call it as what thing? Trachea elements, right? Now coming to the next part, in case of the phloem now. In case of the phloem, to make this diagram more clear, let me to draw separately first of all. This is the upper part of sieve tube. Just now I said you sieve tubes are there, but they are having some pores. They are having some pores at the tip, which you call it as this complete, you call it as what thing? Sieve plate. Having what thing here? Sieve pores. Like we discussed vessel upon vessel, no? In the same here, sieve tube cell is on next sieve tube cell, making a complete sieve tube. Next is there like this. Now one member you call it as sieve tube cell. Fine. And adjoining that is the companion cell. Right? Can you see the companion cell? The word is like a friend. Even this is what cell? Companion cell. Why companion cells are required? Because these sieve tube cells, when they get mature, they will lose its nucleus. They become enucleated. That's why to control its process, they need a company of a cell which is controlling by its nucleus all its function. Are you getting that? So sieve tube and companion cell are the main components of what thing? Phloem. Are you getting that? And sieve tube cells together form what thing? Sieve tube. I repeat, sieve tube cells are going from where? Leaf to the non-green parts of the plant and help in the conduction of the food. But at maturity, they become enucleated. E means what thing? Without. E means without. Without nucleus at maturity, which cells will be there? Sieve tube cells. But don't say they become dead after losing the nucleus. They still are living. Are you getting that? So here the partner of this will be what thing? Companion cells, right? Now coming back to this diagram now. Here what is given? See here, this is what thing? Check my marker where it is going on. See here, this part is what thing? This is a sieve tube cell. And what are these cells? These are companion cells. Then after this is the adjoining one more sieve tube cells and having their companion cells. And this green color is what thing? Parenchyma, which you call it as phloem parenchyma, which is storing the material for this. And fibers either of xylem or of phloem is giving support to these cells. Now can you see these are the different components clustering together but mainly for same function. That's why you call it as tissue. But which type of tissue is now? Complex permanent tissue we had discussed. Are you getting that? Now one more thing we have to go with. What is that? When we discussed about the xylem, xylems can be of two types. What are the two types of xylem? This one. Check it out now. This is important. Xylem, two types of xylem is there. Primary xylem and the secondary xylem. Primary means which is from the beginning. From the beginning when it is there, that is a primary xylem. For your growing plant, they need water and minerals. From the beginning, the xylem which is there in the plant, that you call it as primary xylem. And secondary xylem will form later in the life. Later in the life, when this lateral growth happen, at that time what xylem develop, that you call it as secondary xylem. But which is formed from the beginning, when your seeds sprout a small seedling, that time they are having the primary xylem over there, right? Now see, the primary xylem has two components. One is the protoxylem. Within the primary xylem, the protoxylem will form first. See the word proto. Proto means first. And after change, meta means change. After the change, what is formed, that you call it as metaxylem. Now here the protoxylem will be small in size. It will be small in size, but this will be larger in size, like this. This will be metaxylem, which will be larger in size. Are you getting that? I repeat, xylem are of two types, primary and secondary type. Primary is developed when the plant just grow from the seed, just sprout out. And once it keep on developing until and unless it is not going the lateral growth. Up to that, what xylems are going on forming, they are the primary xylem. And which is formed due to the lateral growth, that xylem you call it as what thing? Secondary xylem, which is forming later in the life of the plant. And these primary xylem, still the xylem has these component, protoxylem and metaxylem. In the same xylem you are having smaller units, means the vessels and tracheids. Suppose this is a vessel, what I have drawn, these are the vessels and the tracheids, which are the pipes going. If I cut it, you will see it like a circle only, no? So that circles I have drawn of what thing? Vessels and tracheids. So that width will be small of vessels and tracheids where in protoxylem, and it will be broad in a metaxylem. but such type of things are not present in case of secondary xylem. I hope so it is clear to everyone. Patches means somewhere, that we have discussed in case of the Collenchyma. It will be some, means at certain places it will be there, that you call it as patches. Fine. Okay, no problem, time is up. Reason is that it is delayed by already 45 minutes this class due to the online problem, right? So I hope so. Can I continue now? Okay, great. So here what is there? Protoxylem and metaxylem, that is in primary xylem. Now coming to the two terms, exarch and endarch. What is that? Now what is there in case of two terms are related to xylem? What is that? New terms, endarch and exarch. Now what do you mean by endarch? Endarch means if suppose this is a plant, right, and central is the part where xylem is there. Fine. The protoxylem, when we are discussing, if it is towards inner side and outer side is a metaxylem, that condition you call it as endarch. Means protoxylem is towards the center of a plant and metaxylem is towards the periphery. Periphery means and the surrounding of that. Such situation you call it as endarch. This is usually the case of stems. In which case it will be present? In case of stem. And if I am discussing exarch condition, in exarch condition, suppose this is a, I must draw here for the roots actually. For the roots, if I am cutting the part over there and protoxylem is towards periphery, now the metaxylem is towards center. This is what thing? Protoxylem. This is what thing? Metaxylem. If such situation is there, such situation you call it as exarch. This is a feature of what thing? Roots. I repeat, see what we have discussed. We have discussed xylem some terms. Xylem is a simple permanent or it is going to be complex permanent? Please give the answer this first of all. Please give the answer to this question. Xylem is a simple permanent or it is a complex permanent? I want your response. I want your attention here, everyone. Very nice. Complex permanent tissue. Are you getting that? And why it is complex? Because it is made up of different elements. What are the different elements over there? Like we discussed, vessel, tracheids, xylem fiber, xylem, parenchyma, right? Now two more terms we discussed for the xylem. It can be primary xylem or it can be the secondary xylem. Secondary xylem is developed later in the life. Primary xylem developed in the beginning. But what they are helping? Both are helping in the conduction of water and minerals. Are you getting that? Now next term we discussed like endarch and exarch. This is very important. Please do focus on these two words because we have to discuss anatomy. There it will help you, right, to identify that this is an anatomy of root or it is of stem. If it is of root, you will find xylem as an exarch condition. I repeat, what is exarch? Where protoxylem is towards outer side and metaxylem is towards the center. That condition you call it as what thing? Exarch, which is in root. Endarch condition where protoxylem is in center, metaxylem toward periphery, and this feature is of stem. Are you getting that? So in that way we have discussed histology. Histology, I repeat, study of the tissues. Now we have to enter into anatomy part. If it is done, it is comfortable, then the anatomy is not going to be tough. We will pick the superficially all the points comparison. Which thing we have to dissect and check the anatomy. First we will take like roots, then we will take the stem, then we will take the leaf. But all the sections we will take for dicot, monocot. Their root, their stem, their leaf. Their comparison we will take. Fine. So on that grounds, let's take that description now. Okay, one more thing I don't want to miss to describe you. What is that part? This thing. Please focus here, all of you. Vascular bundles are what? Which consists of xylem and phloem. Right? Now see, again, what one feature I said you for the root? exarch condition. One more condition is there for the roots, is that radial arrangement of xylem and phloem. When xylem and phloem, they are on different radii. Can you see, this is a circle, these are different radii. When xylem and phloem, they are on different radii, such type of vascular bundle you call it as radial vascular bundle, and this is a feature of roots. R for radial, R for roots. And roots add on even showing which type of condition? Exarch condition. Please keep that in your mind. Radial, I repeat, when vascular bundles or conducting tissues are, I repeat, what are the vascular bundles? Xylem and phloems, which we even call it as conducting tissue. When their xylem and phloem are making the different patches but on different radii, that you call it as exarch, that you call it as radial vascular bundle, and it is present in roots, which is even showing the exarch condition. Now coming to this point, they can even be arranging phloem and xylem in the same radius. If phloem and xylem are on same radius, such arrangement you call it as a conjoint. Co means together, joined together in a one radius. Are you getting? This is a feature of what thing? That is of stems and the leaves. This is a feature of whom? Stems and the leaves. Are you getting that? Okay. So here what is there? The xylem and phloem are on same radius. Such type of vascular bundle you call it as conjoint. Now conjoint has further two types. What are the two types? See here, please all of you focus here. The beginning of the topic I said you intrafascicular cambium. I am applying here now that word. Please see here. I repeat, we are discussing vascular bundles. Which type of vascular bundle I am taking here? Conjoint. Let me take clear color. It is a conjoint vascular bundle. If between phloem and xylem, if cambium is there, cambium is dividing tissue is already present from the beginning, which helps in lateral growth. What is that? I already said you, this cambium is intrafascicular cambium. Intrafascicular cambium. If you cannot understand, at least you should know this intrafascicular cambium is primary meristem, but it is lateral meristem. Primary means it originates from the beginning, it is already there, and this is a feature of what thing? That is in case of the dicot stem. This is a feature of dicot stem. Are you getting that? And if this condition is there, where no cambium, there is no cambium is there, this is a feature of what thing? Monocot stem. This is a feature of monocot stem and even of the leaves. It is not mentioned here, that's why I am writing. Leaves can be monocot or dicot. Both are having closed conjoint. Closed means without cambium. Are you getting that? Whereas here with the cambium, that you call it as open type. This is a feature of dicot stem. This is a feature of monocot stem and the leaves. Both monocot and dicot leaves. Is that clear to everyone? Okay. Now here, tell me your doubt parallelly so that we can discuss that point as well, right? I hope so you are understood by the thing. We are discussing the xylem and the phloem, which you call it as a conducting tissue, or you can call it as vascular bundles. Are you getting that? If vascular bundles are arranged like this, if vascular bundles are arranged like this, like I have discussed earlier, that you call it as radial vascular bundles. Means here phloem and xylem are on the different radii. They are on different radii, that you call it as radial vascular bundle, and if they are on same radius, that you call it as conjoint. Clear? When you cut the transverse section, you will find all these things, but how it is arranged we will still discuss. Just wait for that. Okay, now coming to this part, see here what we are discussing here now, T.S of Root. When we cut the root, see here all of you, your attention should be here. Here is what? Transverse section of root. When you pull out the root from the ground of dicot or of monocot and cut it, you will find like this. You will find like this structure over there. It is a complete circle. It is a complete circle. but we have taken only a part of it. We have taken only a part of it, like you are cutting some pizza, one of the slice out, no, like that. Are you getting? So this root is of what thing? Dicot root. This is of what thing? Monocot root. Fine. Now why I have placed this together? Just to compare it with each other. Like here if I say what is the outermost layer here? What is the outermost layer here? It will be epidermis. What is outermost here? Epidermis. Fine. And to absorb the water, what are there? Root hairs are there. Right, like villi are there, na, I said you in the morphology from outside, the root hairs are coming in the region of maturation. There the absorption is going to be most. If you cut from that region, you will find root hairs. Root hair cannot be on the stem. This is the first difference you can see in the roots and the stem. Is that clear? Okay. Then after this, here what is given? This epidermis is made up of parenchyma cell. What we are discussing, listen this point. We till now discussed types of cells which are in different tissues. Now we are discussing how tissues are arranged to make one organ. So here is one organ in front of you of root. Which root we are taking first? Dicot root. Then we are discussing parallelly to the monocot root. What example here? Like any dicot root, like mustard I can say. Dicot root, and here monocot, like maize I can take an example. We are discussing their root. If you take it out, cut, you will find all these things. The outermost layer is epidermis, which is made up of parenchyma cells. And this all structure you call it as what thing? Cortex. This all cells. Again this is parenchyma cell. Are you getting that? And surrounding by one more layer, inner side, which you call it as here endodermis. Just you should familiar with the terms. Endodermis. It is single, only single. But cortex will be many layered. Can you see? Cortex is going to be many layered. Like brick to brick, cells are arranged making one of the tissues. And what they are creating? The region cortex. Inner to that a circle is there, which you call it as what thing? Endodermis. Are you getting that? And still inner to that one more layer is there, which you call it as pericycle. Just learn the layers. You should aware that where parenchyma is going to be present, where the xylem and phloem are going to be present. So here the epidermis is made up of parenchyma, cortex is made up of parenchyma, endodermis is even made up of parenchyma, pericycle is even made up of parenchyma, and can you see here this part is a xylem part. So it cannot be a parenchyma. Can I call it as a complex permanent tissue? It is a complex permanent tissue now. Which complex permanent tissue it is? It will be xylem. And being this is a root, can I say at this point what will be there? Phloem will be there. Are you getting? And can you see at different radii they have been arranged? So what type of vascular bundle it will be there? Please say that, radial or the conjoint type. As we are discussing the roots, which type of vascular bundle it will be having? Hurry up, please come out with the answers. Conjoint or radial type? Very nice, radial type, very nice, very nice. So here it is having radial type of vascular bundle, where the xylem and phloem are on different radii. Are you getting that? And can you see the center part? This center part is called as pith, which is again made up of parenchyma. And same labeling will go here, endodermis, pericycle, and what is that patch? This patch is of what thing? This is of phloem, and this is of what thing? Xylem. Are you getting that? And here when you will see monocot root, what is a special thing here? What is the difference between dicot and monocot root? I labeled everything all common. For the difference, we will find this diagram. This is your NCERT diagram, right? Labelings already, what I have written, it is already typed here. But what are the difference? Please look here. Here similarities are more, but differences are less. What is that difference? See here. The difference is of, can you see here, xylem patches are how many? Here xylem patches will be 4. How many patches are there? 4 patches of the xylem and phloem will be there. And here how many patches? Many are there. Many patches are there, right, of xylem and phloem. This is the first difference, clear? Second difference is what? Here this pith, the central part, here pith is reduced. Don't write anything. Next slide is having typed differences. Just copy down later on. Just listen the lecture first of all. Here the pith is going to be reduced. But here the pith is going to be developed. In which case it will be developed? In case of the monocot root, right? In case of the dicot root, it is reduced very small. It seems to be same, but it is very much reduced. In which case? In case of dicot root. Then in monocot root, it is going to be well developed. So major are these two differences. Many more are there. Later on we will discuss. But what is the common point? Can I say common thing is both are exarch, fine, and both are having radial type of a arrangement of vascular bundle, and rest all labelings are almost same. Now which cells are parenchymatous, which cells are having the you can say the vascular bundles, or you can say complex permanent tissue, that we have discussed the regions over there. So be focused on the labeling, right? So here we have already discussed this part, and this is telling you about these differences. You please have a look on this later on, but what I pointed out through the diagram, I hope so it is clear to everyone. Okay. So here what we have discussed today, we have to discuss like this part, that is the dicot and monocot root. Just rest of the thing is just for your understanding. If you have understood the root, only our motive is to connect you what thing from the histology to the anatomy part. Histology we discussed complete, that is a main topic, and what else it is focusing, our main heading, anatomy of flowering plant. How the histology is arranged with the different organ, like one organ we have discussed, what is that? Roots. So root of dicot and monocot, that we have discussed. Now just have a superficial idea for the dicot and monocot stem. Just see the differences only. Just have a look that how it appears like this. When you cut the stem of the dicot, it will appear like this. At least have a look on the appearance. We need not to go in much detail right now. But why we discuss the root in much detail? Reason just to say you that how the tissues are going to be arranged. Now can you see a small thing here? Both are stems. In both the stems, there is a conjoint vascular bundles. Are you getting that? Being this is dicot, it is having which type of vascular bundle here? Open conjoint type. Open conjoint type. And being monocot, it is having closed conjoint type. Are you getting that? So need not to worry how it appears and all is going on, but the thing what we have discussed early, just now in my lecture, I said you Collenchyma. O stands for what thing there? Please say me this answer. Collenchyma, what O stands for? Please say here. One student is asking what is secondary growth. Secondary growth means lateral growth like this in diameter. Yes, which is observed under epidermis of which thing? Of the dicot stem. Of the dicot stem. Can I say immediately if this is epidermis, then what is just below it? Just below is what thing? Just below to that is you can say hypodermis, which is made up of which cell? Collenchyma, which is made up of Collenchyma. Are you getting? Here downside epidermis, upper is epidermis and lower is what thing? Hypodermis, which is made up of what thing? This is made up of Sclerenchyma here. Here it is made up of Sclerenchyma. right? Major thing, just have a look over there. I will not go in detail in this. Only in roots we have to go in detail just to introduce how tissues are arranged into an organ. Now here the major difference I said you, immediately after epidermis Collenchyma, here immediately after epidermis Sclerenchyma. Fine. And can you see here vascular bundles are arranged in a ring. Here the vascular bundles, these are all vascular bundles are scattered. What do you mean by vascular bundle? Xylem and phloem, right? So here the xylem and phloem are going to be scattered like a bundle. One after the bundle they are scattered. But here they are arranged into the ring. Only this much. We will not go with the cell to cell, what is the condition over there. But the thing we have to show you when you cut it, how you will see inside that, what view you will look into a stem or in a root. Now last thing we have to go for the today's day, that is about the leaf anatomy. Last is this topic for today. This is a comparison of dicot leaf and monocot leaf. What is dicot leaf? Which is present in dicots, right? Have you seen the grasses? Their leaves are standing straight like this. They are coming out. But when you see the dicot like mango, their leaves are hanging and they are dorsiventrally flattened. Dorsiventrally means upside down like a sticker. It will be like that. But when you will see the grasses, monocots, their leaves are coming out. They are sprouting like that vertically. So that type of leaf here, you call it as isobilateral leaf. Isobilateral leaf is a condition of the monocots, and dicot is having the dorsiventral leaf, right? So here superficially what is the point of difference? Can you see here epidermis in pink color, right? But here the stomata is the spores. I hope so you know the stomata. Stomata are the small pores, like we have nostrils for exchange of gases. These leaves are having stomata for exchange of gases. That stomata number will be more towards lower surface and it will be less towards the upper surface. But here will be equal on both the surfaces. It will be equal on both the surfaces. Are you getting that? I am going only through the important points, and if you find any new term while reading any labeling through these slides later on as a notes, don't be panicked, just mark it, and still we have lot of things to do. This is hardly a summary of the topic we are going with. Fine. So next important thing, can you see these are mesophyll cell, here even mesophyll cells. But here in dicot leaf, these mesophyll cells are divided into like elongated cell, which you call it as palisade cells, and this you call it as spongy mesophyll cells. Are you getting? But here there is no such difference of the palisade and spongy. All are same type of cells in case of the monocot leaf. But here differentiation is there between palisade and spongy parenchyma. Only superficial thing, main you should know about that, right? Okay. This is all about our today's lecture. Now before leaving that, I want to ask you some questions to wind up this session today. Let me to ask you some question. We started from the beginning. We discuss the tissues is cluster of which thing? Will it be of organ or it is of cells? Arya, please give the response. Tissue is a cluster of cells. Very nice, right? And we discussed based on the division, the cells which are keep on dividing, what type of cells these are, which keep on dividing, dividing cells you call it as which cells? The dividing cells you call it as which cell? Meristematic cells. Are you getting that? I should not give you actually open book test, right? Last time I have given you open book test, but not now, right? Already I have given you the hint. Okay, meristematic cell, very nice. Now say me next thing, in xylem, which is the living element? In xylem, which is the living element? Xylem, which is the living element? Arya, please give the response. Meristem is a previous flashing in front of me. Come to the next question now. In xylem, which is a living element, very nice, parenchyma, very nice. Then in case of phloem, which is a dead element, in phloem, which is a dead element, phloem dead element is what? Phloem dead element. No sieve tube, no, it is living. Fibers, very right, it should be fibers. Parenchyma will be always living. Fibers are always dead. Is that clear? So these two categories we had discussed. Now just say me the last question I am keeping in front of you. Exarch condition will be in which case? Root or stems? Exarch. Exarch condition is in roots or the stem? Exarch condition is in root or in stem? Very nice. Not in stem, stem is having end arch condition. Are you getting that? Xylem, phloem, their arrangement, two types we discussed. The radial in case of root, conjoined in case of open conjoined in case of dicot stem. And if I say closed conjoined type, it will be in case of the monocot stem and in the leaves, whether it is a monocot or dicot, right? So don't get worried or tensed by the things, and maybe one go the things are new to you, but if you revise it, just go through the topics gradually, I think so the things will be easier. And don't go with the extra things. If you find in notes, just mark it, but focus on the main points what I discussed with you today. Main is the difference between the root I discussed, monocot and dicot, to link you with the main chapter anatomy. Rest, stem and the leaf, simply I have given the basic differences, right? Just go up to that part. Thanks a lot, student. All the best. God bless you. Thank you.